Okay students, now let us discuss male reproductive system. So here in this male reproductive system, we are going to discuss about the external genital organs and also we will discuss about the accessory glands which are involved in the male reproduction. So here you can see male reproductive system includes a pair of testes, okay, and penis and urethra. So like that these are the three important structures: testis, penis and urethra, and along with that we can see that accessory glands. Okay. So here let us discuss about the testis. Generally if you observe a male reproductive system, generally you can see these are the external genital organs we can see in a male. Suppose you say this is the male body. So in the male body these are the external structure, what you are observing it is called penis, and the structure what you are observing this is a small bag like structure, this is called scrotal sac. So in the scrotal sac, okay, testes are present. Suppose if you see the male animal, so this is the scrotal sac. In this scrotal sac we can see the ball like structure called testis. So like that one pair of testes are located in the scrotal sacs. Generally this reproductive system is hanging outside the body. Why it is outside? Actually you see the scrotal sacs, they are outside. So why? Because 2 degrees centigrades to 2.5 degrees centigrades less temperature is present. So if it is inside the body, there will be 37 degrees centigrades of body temperature, but here in scrotal sacs only 35 degrees centigrades are there. It is less than the body temperature, that is 2 degrees to 2.5. So this is suitable for the spermatogenesis process. For the production of sperms, this temperature is suitable. So that's why they are coming out from the body. So in the scrotal sacs we can see the testis, and suppose if male animal is running or male animal is jumping, the testes are protected and prevented dislocation by gubernaculum. What is that? Gubernaculum, which is giving support from the posterior side, and anterior side of testis you can see it is a spermatic cord. So like that spermatic cord is giving protection from the anterior side, gubernaculum is giving protection from the posterior side. So like that testes are placed in the positions in the scrotal sacs. So like that this type of arrangement is there. You know, this is the penis, which is the intromittent organ useful for sexual intercourse. So like that these are the external structures we are observing in a male. In a baby, if you see, in a baby testes are actually located in the abdominal region. So in the embryonic condition testes are located in the abdominal region. So when the embryonic development is completed, so the baby is ready to come out from mother womb. So during that time generally these testes are coming back to the original positions, that is called scrotal sacs. So scrotal sacs. So here the testes are coming from where they are coming? They are coming from abdomen. Where they are coming? They are coming into scrotal sacs. What is this canal? This canal is called inguinal canal. What is this? Inguinal canal. So through the inguinal canal testes are placed in the scrotal sacs. So just before the delivery we can see that. So after completion of embryonic development, when fully grown fetus, పూర్తిగా అభివృద్ధి చెందినటువంటి ఫీటస్లో అబ్డామెన్ నుంచి ఇంగ్వినల్ కెనాల్ ద్వారా స్క్రోటల్ సాక్లోకి టెస్టిస్ రావడం జరుగుతుంది. ఒకవేళ కొంతమంది పిల్లలకి అలా జరగదు. అలాంటి కండిషన్ని క్రిప్టార్కిడిజం అంటారు. వాట్ ఈస్ దట్? క్రిప్టార్కిడిజం అంటే అబ్డామెన్లోనే టెస్టిస్ ఉండిపోవడం. సో రిటైనింగ్ ఆఫ్ టెస్టిస్ ఇన్ ద అబ్డామెన్ ఈవెన్ ఆఫ్టర్ బర్త్ ఆల్సో. that type of condition is called cryptorchidism. This is going to be rectified by a small surgery. Because of that, again they are placed in the scrotal sacs. So, let us move to the structure, how testis is present inside the, scrotal sacs. We will see now. Suppose if you see, this is a scrotal sac, it is completely zoomed diagram. So, here you see, this is the structure, already this is about spermatic cord, which is giving support from anterior side of the testis, and just now we discussed about this posterior part. What is this? It is called gubernaculum. It is gubernaculum, and testis is covered by outer envelope is called tunica vaginalis. What is this? Tunica vaginalis. So, this is tunica vaginalis, which is called outer envelope, and the pinkish color testis, if you see, this is immediately covered by tunica albuginea. So, testis is immediately covered by tunica albuginea. So, this tunica albuginea is going to divide the testis into septas. So, like that, more than 200 septas you can see. So, inside the testis there is many septas are there, that is more than 200, like that up to 250, that is done by the tunica albuginea layer into septas. In each septa, you see this, in each septa there is coiled tubes, 2 to 3 coiled tubes. How many? 2 to 3 coiled tubes are there. These coiled tubes are called, seminiferous tubules. What are these? These are called seminiferous tubules. So, like that, seminiferous tubules are present. In each septa there are 2 to 3 seminiferous tubules are present. Let us zoom the seminiferous tubules inside the septa, how it is, we will see now. Generally here, suppose if you see the septa inside, suppose this is the septa you are observing in this. Let us take only one pipe. Just now I told there is 2 to 3 seminiferous tubules are there. One seminiferous tubule you are observing inside the septa. So, that red color border is septa, inside that seminiferous tube. So, in the seminiferous tubule, sperm mother cells are there called spermatogonia. Spermatogonia are sperm mother cells. Not only that, we can see here and there Sertoli cells also present. They gives nourishment to the sperms. Sertoli cells. So, like that, inside the tube, inside the lining, you can see two types of cells. One are spermatogonia, another one Sertoli cells. Spermatogonia gives birth to sperms. Sertoli cells gives nourishment to the sperms. So, like that, the two cells are there. Again, in between the tubular gaps, so in the interstitial spaces, you see here, so either side of the tubes, you see in this space, you see this diagram clearly. So, here and there in the gaps between that, so in the interstitial space, there are special type of cells are called, they are called Leydig cells. What are these? Leydig cells are present, they are called interstitial cells. They are very important. Why? Because they secretes testosterone. What is the hormone? Testosterone is a male android hormone, that is androgen, which releases testosterone, which is very much useful, which will change the environment, which promotes the spermatogenesis process by changing the environment. So, like that, so total 3 types of cells we discussed. One is Leydig cell, spermatogonia and Sertoli cell. So, like that, sperm production environment is created by Leydig cells, spermatogonia gives sperms, Sertoli cells gives nourishment. So, like that the cells are there. And next step, if you see here, all the spermatogonia, okay, which produced sperms, all the sperms through these tubes, see, all are united in one place, right. So, this is the junction. You can see that it is a rete testis. What is this? Rete testis is a junction which is gathered, okay, gathering of all the seminiferous tubules. So, all the seminiferous tubules, you see this junction, this is called rete testis. From this rete testis, you see some small tubes are coming out, they are called vasa efferentia. What are these? Vasa efferentia. So, like that, vasa efferentias are coming out like this from rete testis. So, seminiferous tubules, rete testis, vasa efferentia. So, like that, sperms are coming, and finally all the vasa efferentias are going to give a common tube and highly coiled tube. This highly coiled tube is called epididymis. What is this? Epididymis. Epididymis is a highly coiled tube. That highly coiled tube is going to give a very big vasa difference. Before that it was vasa efferens. So, seminiferous tubules, rete testis, vasa efferentia and epididymis. Okay students, you see the transportation of sperms here, rete testis to it is moved to vasa efferentia. So, see this vasa efferentia. So, finally it is united to form a highly coiled tube. You are observing like this fully coiled tube, that is caput epididymis, corpus epididymis, cauda epididymis. This is one of the important part in male reproductive system, because the sperms will come and take rest in the epididymis, and not only takes rest, sperms will become mature in that area, and also sperms will get nourishment also in that place. So, that is why epididymis is very important, that is going to divide into caput epididymis, corpus epididymis, and the last part is called cauda epididymis. This cauda epididymis is elongated as vasa deferens. So, here the single tube is called vas deferens. So, like that epididymis continued through the spermatic cord, you know already. So, in the spermatic cord which is travelling, vasa deferens is travelling. Not only vasa deferens, nerves and blood vessels are also present in the spermatic cord. Nerves and blood vessels and vasa deferens and connective tissue together called spermatic cord, because through that there is a connection towards abdomen. So, vasa deferens which is passing towards the abdomen, and where it is going to open, let us see now. So, whatever the vasa efferentias you observed, that vasa efferentias are together formed epididymis, that epididymis continued as a vasa deferens. Now, just a previous diagram you saw that vasa efferentias are moving towards abdomen. So, these vasa deferens, they are moving in front of the urinary bladder, means ventral side to the urinary bladder, ventral side to the urinary bladder. You are observing the, okay, the tubes which are went towards abdomen, they are again coming back. So, like that the vasa deferentia tubes are going to get connection with a gland called seminal vesicle. You see I wrote here SV, means this is seminal vesicles. So, like that vasa deferentias are going to connection with, what is this? Seminal vesicle. So, this is vasa deferens and this is seminal vesicle. Two tubes together forms a common tube called ejaculatory duct. Vasa deferens and duct of seminal vesicle together forms a common tube. This common tube is called ejaculatory duct. You see this one pink and you see this one black. So, both are united here. So, together forms ejaculatory ducts. So, like that two sides, two ejaculatory ducts are there. Two sides, two ejaculatory ducts are there. Here the importance of seminal vesicles are, the seminal vesicles provides semen, that is almost 60% of the semen is produced, which consists of fructose, prostaglandins and the important substances which are very much useful for the maturation and also giving energy to the sperms. Next, ejaculatory duct. You see where the ejaculatory ducts two are there, where the two ejaculatory ducts are united, that particular area, neck of the urinary bladder. So, that neck of the urinary bladder, that is called urethra. So, in that region, below that urinary bladder, in front of that means from ventral side ejaculatory ducts. So, urinary bladders opening and this ejaculatory ducts union together forms, around that there is a single prostate gland is present. So, this is one prostate gland. So, here you see a pair of seminal vesicles you observed before that. Now, you are observing a prostate gland, it is like a band around the urethra, that is very important because it will secrete 30% of the semen. So, this is also very important, that which can provide nourishment to the sperms and activation of sperms. And the next one you can see this channel, here you see 𝗂𝗽𝗱 gland. What you are observing? 𝗂𝗽𝗱 gland. Below the prostate gland, one pair of 𝗂𝗽𝗱 gland you are observing. So, like that seminal vesicles one pair, prostate gland one, 𝗂𝗽𝗱 gland two. So, like the two 𝗂𝗽𝗱 glands are present below the prostate. It is also very important, it is called flushing agent. Why? Because see, this is the pipe from where the urine is passing, right. So, this is urethral, urinogenital duct. The duct is passing to the penis, from the penis it is going out as a glans penis and urethral orifice. So, like that the pipe which is coming over here, either side of this you can see the 𝗂𝗽𝗱 glands. If there is any urinary residue, means any urinary drops are there in this tube, they are flushed out by the secretions of 𝗂𝗽𝗱 gland. So, like that they neutralizes this passage. So, that is the importance. Okay, coming to the last part, here you see the hanging structure, cylindrical rod like structure, that male external genitalia, that is called penis. So, if you see the penis structure, generally penis is rod like structure which is with you can see that. So, this is with the tip, that is called glans penis, tip is called glans penis. So, this glans penis, if you see, it is covered by a loose sheath, this is called prepuce. What is this? Prepuce. So, like that prepuce is a layer which is a loose layer and glans penis is a tip of the penis. Inside the penis, if you see, there is a three muscular bundles are present. Okay, what are that? Inside the penis, if you see the transverse section of penis, you can see corpora cavernosa. What is this? Corpora cavernosa. That is a sensitive tissue which is very much useful for making penis strong and long at the time of sexual excitement. So, like that here in the penis it is going to become strong and long, and that is called erectile condition, that is because of the tissue that is called corpora cavernosa. Total how many bundles of corpora cavernosas are there? 2. And the another bundle you can see corpora spongiosum, corpora spongiosum. So, here in the corpora spongiosum, the urinary orifice, urinary opening, that is called urinary orifice is present. So, that is called urethral orifice. Why? Because urinogenital duct. So, urine and as well as semen both are coming. So, that is why we can call it as a urethral orifice. So, here urethral orifice is useful for releasing both semen and also urine, but both are not at a time. So, regularly the urine will pass through the tube. So, and we can see according to the time the semen will also pass through that canal. So, like that this is the male reproductive system, and 3 varieties of accessory glands also we discussed in this chapter. And also this diagram is very important in the competitive exams. Sometimes they will give you the diagram and ask you about the labeling, or else what is the importance of particular structure. In the competitive exams you have to go through the all points related what I teach you now. Students, let us discuss female reproductive system. So, here in female reproductive system, along with the reproductive structures, that is called external genitalia, and also we will discuss the structures which are supporting to female reproductive system along with accessory glands. So, now let us move to the structure. Suppose if a lady is moving in this way, so she is standing over like this. So, here you can see in this diagram, urinary bladder is first, then uterus, then you see rectum and anus. So, like that, urinary bladder, uterus are together independently opening in a slit like structure called uvula. So, like that, in female, urinary bladder, uterus both are opening in a slit like structure called vulva, okay, or which is also called pudendum. So, then behind that, just after that, you can see another hole, that is called anus, which is the ending of that rectum. So, like that, in female reproductive system, urinary orifice and vaginal orifice, two different openings are there in the vulva region. But in male reproductive system, it is not like that. Urinogenital, there is only one opening is there for both releasing of semen and urine. But in case of female, it is not like that. There is two separate openings are there, urinary orifice, that is related to urinary bladder, vagina, that is related to female reproductive tract. So, like that we can see here. And one more important thing is that in male reproductive system, first male reproductive system is there in the ventral side, then okay, urinary bladder is there. But here urinary bladder is there in ventral side, then uterus is there. So, which is in the ventral side, urinary bladder is in the ventral side, then behind that you can see the uterus. So, like that, urinary bladder and okay, uterus we can see together, okay. And next, coming to the only reproductive system of female. So, here here you studied about only the arrangement, but here you are going to see the complete structure of female reproductive system. See here, it starts with vagina and the neck of uterus is called cervix. Neck of uterus is called cervix, or else we can say that entrance towards uterus is called cervix. Then you can see a very big bag like structure, that is called uterus, and you see there is a two tubes you are observing that are called fallopian tubes. Two fallopian tubes united to form a very big bag like structure called uterus, or else we can call it is a womb. Then you can see that in red color I used here for the solid mass of structures, these are called ovary and ovary. So, together ovaries. Total how many ovaries are there? 2 ovaries are there. So, like that, in a female, ovaries, fallopian tubes, uterus and vagina, these are the female reproductive organs. So, let us discuss one by one according to the diagram we will go. First we will take ovaries. First, if you take the ovaries, what is going to see we will now. See, ovary structure, if you see, it is divided into two regions. Ovary is covered by tunica albuginea. Ovary is covered by tunica albuginea. So, here in this tunica albuginea, outer cortex is called cortex region. Outer part is called cortex region. Inner part in the ovary is called medulla region. So, what we are discussing now? We are discussing about one ovary. In one ovary, outer cortex is there, inner medulla is there. It is fully made with germinal epithelium, especially cuboidal epithelium is present, which produces ova. So, here in this cortex region is there. Cortex region shows many different stages of developing eggs. So, in female ovary, if you see this ovary, inside we are observing, we are zooming this. So, cortex region is the outer part with so many stages of developing eggs we are observing. Medulla region you are observing, which is in the center, which consists only blood vessels and nerves and connective tissue. Blood vessels, nerves and connective tissues are present, right. So, what are present in the cortex region? That is called primary follicle. What are these? Primary follicle or else secondary follicle. Okay, different stages. secondary follicles, or else final stages that are called Graafian follicles. What are these? Graafian follicles. What is the importance of Graafian follicle? If you take a Graafian follicle, so Graafian follicle is very important follicle. If you observe this Graafian follicle, which is surrounded by, okay, so lot of many layers called theca. What are these? This is about theca externa, theca interna. Theca layer, what you are observing, it is especially theca interna releases estrogen hormone. So that's why from the ovary, theca interna is very important because it secretes estrogen hormone. And not only that, there is a space is there, that space is called antrum. And corona radiata, zona pellucida, CR means corona radiata, ZP means zona pellucida. So like that, corona radiata, zona pellucida, and inside you can see the secondary oocyte. So what is present in a Graafian follicle? In Graafian follicle, secondary oocyte is present. What is present? Secondary oocyte. So like that, okay, theca interna, theca externa, first theca externa, then theca interna, then antrum, then cumulus euphorus, then corona radiata, then zona pellucida, then secondary oocyte. So like that, these type of fully developed Graafian follicles we can see in the ovary at the time of ovulation. So like that, once it is ovulated, for example, this Graafian follicle become empty. Why it is become empty? Because after ovulation, this Graafian follicle is called empty follicle. Okay, so fully developed secondary oocyte along with corona radiata, zona pellucida came out. That is called ovulation. So after ovulation, what happened? This empty follicle is going to turn like yellow mass. This is called a yellow mass. That yellow mass is nothing but corpus luteum. What is this? Corpus luteum. So this corpus luteum starts producing progesterone hormone. What is that? Progesterone hormone. That progesterone hormone, whatever is releasing, that maintains endometrium. So why? Because whatever the ovum is released, that is called secondary oocyte is released, suppose if it is fertilized with sperm, so there is a space and also there is a implantation is needed for the fertilized ovum, that is called zygote. So like that, endometrium wall is prepared. Okay, so that is for that purpose progesterone hormone is secreted by corpus luteum. Suppose if there is no fertilization, so the secondary oocyte become waste. So automatically, so this corpus luteum is converted into, corpus luteum will converted into a white color scar called corpus albicans. What is this? Corpus albicans means corpus luteum become vestigial and finally shrink into a white color structure, that is a scar called corpus albicans. So like that, inside the ovary you can see that every month, especially in the females, okay, in the middle of the menstrual cycle, that is almost in the 14th day of menstrual cycle, ovulation occurs. So like that, Graafian follicle, from Graafian follicle, so ovary is released out, that is called secondary oocyte. So corpus albicans, corpus luteum, corpus albicans, corpus luteum, corpus albicans, like that stages we will observe continuously. Okay, if she becomes pregnant, so there is no need of corpus albicans, corpus luteum will continue for 4 months of the pregnancy, remaining that placenta will take care about the pregnancy. But up to 4 months, corpus luteum takes care about the pregnancy by releasing progesterone hormone. Okay, so if fertilization does not occurs, what happens? Corpus luteum is going to become corpus albicans. So like that, ovary plays very important role during the okay, fertilization and also implantation. And another thing we are going to discuss about the tubes. Okay, let us move to this tube. You know what is this tube? What is this? Fallopian tube. Yes, if you see the, now let us discuss fallopian tube. So here the fallopian tube consists three main important regions called infundibulum, ampulla, isthmus. Infundibulum, ampulla, isthmus. So like that each tube consists three regions. You see, first infundibulum is a funnel like structure with finger like edges, right. So what is the use of these finger like edges? You know, they are called fimbriae. These fimbrias are very much useful to capture the secondary oocyte. At the time of ovulation, from the ovary, secondary oocyte is released, that is captured with the help of fimbriae. So like that the finger like structures are called fimbriae and the infundibulum, that is very important to receive secondary oocyte. Then, so if you see this inside the lining, if you observe, what is the specialty of this fallopian tube? If inside you can see ciliated columnar epithelium is present. What is that? Ciliated columnar epithelium. Very important point. This ciliated columnar epithelium, cilia will move like this. So the movement of the cilia which are going to pull the secondary oocyte towards ampulla. So like that in the ampulla, secondary oocyte is waiting. Why? Because during the sexual intercourse, if sperms are entered, so that sperms will travel through the fallopian tube and they will reach the ampulla, and in the ampulla region fertilization occurs. So like that, so from here, from fimbriae to infundibulum to ampulla, the travelling of ova you can see. So with the help of columnar ciliated epithelium, simple ciliated columnar epithelium is very important to push the ovum towards ampulla. Okay, suppose if it is fertilized, then the zygote is also transferred through isthmus to uterus. So like that, this is very important. From here ovary, secondary oocyte is released, here in the ampulla region it is fertilized, and the fertilized zygote is passing through this channel. Fertilized zygote is passing through the isthmus. Yes, fertilized zygote is passing through the isthmus and finally coming into the uterine region. So at the stage of blastocyst, which stage? Blastocyst stage. Okay, at the stage of that blastocyst, okay, we can also called embryonic disc, that blastocyst stage will come and settle here. So it will travel like this and finally it is going to get implanted with the uterus. So let us see uterus. So here you see in this diagram, suppose if you observe the wall of uterus, it is made with 3 layers. So now I am going to discuss about uterus. So how is uterus? Uterus is made with 3 layers. So like that, okay, so this is 3 types of layers now we are. So let us see the structure of uterine wall. Uterine wall is made with 3 layers. The outer layer is called perimetrium. Outer layer is called perimetrium and the middle layer is called myometrium. So this myometrium layer is very important because it is made with smooth muscles. It is made with smooth muscles. So that's why it is very important, because at the time of okay, child delivery, so the contractions are very important to the myometrium. And the another structure, endometrium. Which one is the lower layer? Endometrium layer is the inner lining of the uterine wall. So like that, perimetrium, middle myometrium, inner endometrium. So like that 3 layers are there, but the endometrium layer is not so constant. Why? Because suppose if the ovum whatever is released, if it is not fertilized, if the ovum is not fertilized, there is no need of endometrium. Why? Because endometrium is very much useful for implantation. So the embryonic development will occur attached to the endometrium, but here the endometrium is no need. Why? Because there is no fertilization. So during that, okay, endometrium wall is destructed and released out through menstrual bleeding. Suppose if fertilization occurs, the endometrium wall will going to get fully developed, which can able to hold the developing embryo perfectly. So that is the role, that is reconstruction and destruction of endometrium regularly we will see during the menstruation in females. So this is the about uterine wall. Now let us see the structure which is called pudendum or vulva, which is a slit like structure in the female external genitalia. If you observe this structure, okay, so it is a slit like structure. Here you can see two fleshy lobes are there. So they are vertical lips, they are called labia majora. What are the vertical lips in the female reproductive tract? They are called labia majora, and above the labia majora you can see fat is observed. So it is a fleshy fat with hair. That particular structure is called mons pubis. So mons pubis, labia majora, almost together you are observing here, and inside the labia majora another very thin lips are present. You see in red color I mentioned here, these are called very thin lips which are inside the labia majora you are observing. That foldings are called labia minora, labia minora. So like that labia majora and labia minora, two structures we can see inside the vulva. And see, there are two openings you are observing. Just in the introduction I told, female vulva consists two independent openings, one is for urine and another for female reproductive system. So like that you see here, this is called urethral orifice. So this urethral orifice, this structure is called urethral orifice. So this urethral orifice is useful for releasing urine, and below the urethral orifice you are observing a wide opening. That wide opening is called vaginal orifice. So this is the vaginal orifice. If you compare the diagram here, so this is the vagina, here you are observing that, and in the female around the vagina you can see there is a very thin layer is there, that is called hymen. So a protective layer around the vagina is called hymen layer. So like that vagina is going to get enter into the cervix, from the cervix it is going to connect it to the uterus. And the one more important structure is there, this is called clitoris. You see in the upper corner above the urinary orifice and below the upper corner of the labia minora you can see a small rod like structure, that is called clitoris. It is a sexually excitement organ in the females, and also it is equal to male penis. Why? Because in male penis we studied about corpora cavernosa. Same like corpora cavernosa tissue we can see in this clitoris region. So that is why it is equal to penis, and which is very much useful for the sexual excitement in the females. So like that this is the structure we can see in female as vulva. Now let us discuss accessory glands in female reproductive tract. So here you see in the vagina, corners of the vagina if you see, there is two glands are there. The major important glands are Bartholin's glands. What are the important glands? Bartholin's glands are very important glands. They are present in the lower corner of the vaginal orifice. Bartholin gland secretions are very important. When the female gets sexual excitement, at that time it will release mucus and lubricating watery fluid, which is very important for the penetration of penis into the female body. So like that Bartholin glands allows the penis into the vagina by lubricating the vaginal passage. They are equal to bulbourethral glands in male. They are equal to bulbourethral glands in male. And you see another glands, they are called Skenes glands. On the anterior margin of the vagina you can see, anterior margin of the vagina you can see the Skenes glands. What are that? Skenes glands. Generally they will neutralize the urinary residue. So and also they are very important for the activation of sperms inside the female reproductive tract. They are equal to prostate gland in male. They are equal to prostate gland in male. So like that only two varieties of accessory glands we can see in female reproductive tract. So here you have to draw the diagram perfectly, you have to label the parts perfectly, and also you have to know very well about these particular parts, and in the examination point of view this is the complete diagrammatical pattern. So, that is why complete anatomy is very important in the competitive exams. So, any part they may ask from this particular topic. Okay students, let us discuss embryonic development. So, here in the embryonic development you see how from fertilization onwards to the complete development of embryo to fetus and childbirth. So, what are the events? So, generally it is called embryology. So, study of events from fertilization to complete development of embryo and also becoming into fetus, these all the events we are going to study in this embryonic development. It is nothing but embryology. So, here we can see once sperm unites with the secondary oocyte and finally with ovum and forms a zygote, right. So, after that in the zygote, so after fertilization this zygote, whatever the zygote is there, that undergoes cleavages. So, once it is a single cell, that is diploid zygote, that is undergoing cleavage in radial manner. So, that is why you can see holoblastic cleavages, radial indeterminate cleavages we can see, radially cleavages will be there, and the cleavage cells, whatever the cells are there, their fate is not yet decided. So, that is why they are called indeterminate cleavages. So, like that entire zygote is going to get cleaved. Why? Because microlethal egg, there is no yolk in the ovum or there is no yolk in the zygote. So, automatically what happens? There is no yolk. So, that is why completely that zygote undergoes cleavage. So, that is why it is called holoblastic cleavage, because the reason is microlethal, or we can also called alethal egg. Microlethal egg means very little yolk is there. Alethal egg means there is complete absence of the yolk. So, mammalian eggs are especially in eutherians, they are alethal. Okay. So, radial indeterminate cleavages occurs after fertilization. After fertilization 36 hours after, this point is very important, after 36 hours of fertilization the cleavages occurs. Okay. Finally, so many cleavages occurs. Once it is going to get 2 cells, 2 cells will become 4 cells, 4 cells will become 8 cells, 8 cells will become 32 cells, 32 cells will become 64 cells. So, like that doubling number you can see in the cleavages. Finally, there is a solid mass of mulberry, okay, marula. So, it looks like mulberry fruit. So, that is why it is called mulberry marula. So, fully packed, tightly pressed with each other. So, this type of pressing the cells and binding together, so many cells are cleaved, the size is not increasing, the very small size cells are, okay, small sized cells you are observing inside that zygote, the size is not increasing about that particular zygote is converted into marula now, but the cells are very small, they are tightly placed with each other, that is why this condition is called compaction. Okay, because of the compaction you can see inner cells are coming together and outer cells are going to get separated as a outer protecting layer. So, inner cells are becoming together, outer cells are becoming a outer protection layer. So, this outer protection layer is called trophoblast layer, trophoblast layer, and the inner cells whatever you are observing which are with the compaction process tightly pressed with each other, they are called embryonic mass, they are called embryonic mass of cells. Okay. So, like that the main embryo is going to get from that embryonic mass, trophoblast is the layer which gives protection. So, like that stage you are going to see. Then afterwards what happens? Okay, this stage where you are observing this stage is now started moving from ampulla to, so where it is actually happening in the fallopian tube it is there. Why? Because you know in the ampulla region fertilization occurs in the fallopian tube. So, like that here it is slowly gradually started moving towards isthmus. Where it is coming? It is moving towards isthmus and finally it is going to get into uterus. So, before going to get into uterus there will be a cavity development inside. So, like that this is the outer layer called trophoblast, you know very well, here you are observing, and this is about inner cell mass or embryonic disc. So, inner cell mass or embryonic disc is here. So between what you are observing? Between you are observing a cavity. This cavity is with fluid. So this process is called cavitization. So like that, cavity you are observing, a cyst like condition, a bag like structure you are observing. Inside the bag you are observing the embryonic mass. So this is called blastocyst stage. This is called a blastocyst stage. So this blastocyst stage is going to get into uterus, and before that it is received fluid from the surrounding area. So now it is a fluid filled cavity. Now this stage is called blastocyst stage. Now let us see the implantation. Why? Because now you see from here to here, you know, already from ampulla to isthmus, already the blastocyst is passed, right? So it is coming towards uterus. So once it is entered into the uterine cavity, the zona pellucida layer, the outer embryonic, that layer, you know already, zona pellucida layer, that is going to get dissolved with the help of trypsin enzyme. What is that? Trypsin enzyme is released by the trophoblast, and finally that zona pellucida released. From that, along with trophoblast, this embryonic pole comes out. So like that, the envelope of zona pellucida is cleared. Now trophoblast, now the zona pellucida layer is dissolved, along with trophoblast, embryonic disc and blastocyst released, become free. So right, this trophoblast layer releases some proteolytic enzymes. So because of that proteolytic enzymes, it is going to get and fixed with endometrium. So this is called implantation. What is implantation? Tell me. The blastocyst. What is this? Blastocyst stage, especially the outer layer of blastocyst. What is the outer layer of blastocyst? Trophoblast. So trophoblast releases some proteolytic enzymes that dissolves the endometrium of uterus, and it is going to fix with the uterus. So implantation stage is very important. After 6th day, and almost in the 7th day or 6th day, you can see the implantation with the uterus. Okay. So what is going to happen to that? We will see now. See, this is embryonic pole. What is this? This is embryonic pole. From this embryonic pole, one layer of cells are coming out. You see this? One layer is coming out by leaving some space. So this is called hypoblast. What is this? It is called hypoblast. So like that, this process is called delamination. After implantation, what happened? Delamination. So here first we saw compaction, later we saw cavitation, then now we are observing implantation. After implantation, what happened? After implantation, from the embryonic pole, one layer of cells are moving towards the trophoblast by leaving some space. These type of cells are called, this type of layer is called hypoblast. This process is called delamination. So like that, you can see one side hypoblast, another side embryonic pole. So like that, bilaminar embryo you are observing here. After that, if you see only the bilaminar embryo, you know this is now named as epiblast, and this layer, what the layer which came out, now it is called hypoblast. So like that, hypoblast comes out leaving the group together, that is called epiblast. This epiblast is very important. Why? Because from this epiblast, amnion layer will come, and also not only amnion, 3 germinal layers, ectoderm, mesoderm, endoderm. 3 germinal layers also comes out, and a full-fledged embryonic formation we can observe from the epiblast. Hypoblast is only extra endoderm layer, that is not that much important in embryonic development, but epiblast is very important. From epiblast, how mesoderm will spread, how endoderm will spread, now let us see. Let us discuss. So how the 3 germinal layers are going to get separated. Now in the gastrulation stage we are discussing. Just now we discussed epiblast and hypoblast formation. So after that, you see in the gastrulation stage, migration of cells. So in the gastrulation stage, what happened? Migration of cells are there, especially the movement, rapid movement of cells we are observing in the gastrula stage. So, what is going to happen, we will see in this diagram. You see, this is epiblast with outer layer, but inside you can see the mesodermal migration you are observing. So, once the mesoderm is going to get, mesodermal cells are going to get migrated, at that time you can see a line. See, what is this line? This is called primitive streak. So, this is the first identification character of gastrulation and formation of mesoderm. So, the cells which are mesodermal, they are migrating all over, which leaves a primitive streak, and either side you can see primitive folds and primitive groove appeared on the surface of epiblast. Then the anterior parts of that primitive pit, okay, before that primitive knob, or we can call Henson's node. So, like that primitive streak, primitive pit, primitive knob, like that structures are formed on the epiblast as a line. So, below that what is happening ante mesodermal migration is happening. So, like that ectoderm, mesoderm and endoderm, 3 layers are there. So, ectoderm is as it is, mesoderm is showing migration. So, because of this endoderm gets separated, separation of this endoderm is very important. So, this process is called ingression. What is ingression? Ingression means from epiblast endoderm layer is separated. The process of separation of endoderm from epiblast is called ingression. So, like that towards hypoblast, the original endoderm is moving, leaving the space, right. So, after that how mesoderm will occupy the entire region, we will see now. So, mesoderm initially moves and forms the first organ in our body, that is called notochord. So, what is that? Notochord. So, from notochord, that is very important structure, from mesoderm, chorda mesoderm, a solid structure notochord is formed. This is the first structure we can notice in the embryonic development. So, this notochord stimulates the ectoderm to form nerve cord by the process of induction. What is that? Induction. So, first from mesoderm notochord is formed. Above the notochord there is a ectoderm called neural plate. From that neural plate nerve cord is formed by the process of induction. So, like that notochord and nerve cord structures you are observing, and here this is the structure from endoderm it is formed, that is alimentary canal. So, remaining areas are fully occupied by mesoderm. So, like that mesodermal domination you are observing as 3 units. One somite you can see epimere, that is present either side of nerve cord and notochord. Another somite you are observing, that is called mesomere, which is in the middle, and another one hypomere, which forms cavities. So, epimere is a solid structure, mesomere is also a solid structure, but hypomere is not like that, it is a cavity. So, here epimere gives sclerotome, dermatome, myotome. From epimere, sclerotome means here connective tissues. What are coming? Connective tissues are coming. From dermatome, skin is coming. Sclerotome gives vertebral column. What are these? Vertebral column, that is bone. That is, next dermatome gives skin and connective tissue. Skin and connective tissue. Myotome gives muscles. Myotome gives muscles, especially skeletal muscles. So, like that vertebral column, skin, connective tissues, muscles are developed by epimere. The next mesomere. You see mesomere forms urinogenital system, urinary system, reproductive system, together called urinogenital system. So, that is why you see in male reproductive system and female reproductive system it is associated with excretory system. So, like that urinogenital systems are derived from one type of tissue, that is from mesoderm, that somite is called mesomere. Next one is hypomere. You see the hypomere, you are observing hypomere is a cavity layer because which forms coelom and also which forms blood circulatory system with open tubes, that is about blood vessels like arteries and veins, because the cavity is needed for the flow of blood in the arteries, veins and capillaries, together forms a closed type of blood circulation. So, like that here hypomere shows 2 layers. One layer is outer layer somatic peritoneum, which is near to the body wall, near to the gut, this is called visceral peritoneum. Somatic peritoneum, visceral peritoneum, like the 2 layers are there, outer layer is somatic peritoneum, inner layer is called visceral peritoneum. Between the two layers we can see the coelom. Now let us see the extra embryonic layers which are going to protect the baby, that is called fetus especially. So now we are going to discuss about the extra embryonic layers which are going to give protection to the baby. So yes, you see here this is the embryo, or you can see the fetus. So here this is called amnion layer. What is this? Amnion layer, the complete layer you are observing around the embryo, which consists amniotic fluid, amniotic fluid. So like that, this amniotic fluid which protects the baby and prevents from desiccation and malformations. So like that, this is one of the important layer. Along with the fluid, it is very important. It is derived from epiblast. So like that, amnion layer you are observing. And another layers you can see, this is called chorion. This is completely outer layer. What is the complete outer layer? Chorion layer. This chorion layer consists many foldings. They are called chorionic villi. Why? Because these chorionic villi are going to get connected with endometrium, of uterus. So like that, with the uterus, with the help of villi, chorion layer is going to get connected. So like that, chorion is the outer layer, and immediate to the embryo you are observing amnion, and this is the layer you are observing yolk sac, and this is the layer is called allantois. What is this? Allantois. So like that, the inner layers you are observing yolk sac and allantois. Yolk sac is connected with the so umbilical cord we can also call. This umbilical cord is connected to midgut of the embryo, that is middle of the alimentary canal. And another one you can see hindgut region. So nearly the anus region, hindgut region is going to connected to the allantois, so which can able to store the fecal matter and also helps in respiration. So here you can see allantois also have villi. Chorion is showing villi, allantois, both are united. Here you can see this particular part. Here chorion is there, here allantois is there. So that's why human placenta is called chorioallantoic placenta. So like that, chorion and allantoin very important for the transfer of nutrients. And also here near the yolk sac you can see there is a umbilical artery and umbilical vein. Umbilical artery and umbilical vein, they are also connected with the middle of the alimentary canal of the embryo. So like that, these are the four important layers: chorion, amnion, yolk sac and allantois. Here yolk sac is not that much useful. Why? Because nutrients are get supplied from the mother blood circulation. That's why in the humans yolk sac is not useful, but the umbilical cord and umbilical veins are connected through the midgut from the mother. Let us discuss some events as a timetable of this embryonic development. You see, that is total 9 months of pregnancy period is there for human beings. So among these 9 months, if you make it into 3 trimesters, means 3 months first trimester, next 3 months another trimester, last 3 months another trimester. So like the total 9 months are divided into 3 trimesters. So first trimesters we can use the word embryo, remaining 2 trimesters you can use the word fetus. So like that, embryo and fetus, like that you can call. First month, if you see the first month, there will be a development of heart, and not only development of heart, it also starts heartbeat. So by listening with the stethoscope, we can hear the sound of fetus heartbeat at the age of 1 month embryo. And at the end of second month, you see end of second month, you can see the limbs and digits are formed. Small size limbs, hands and finger digits formation we can see in the end of at the end of second month. And end of the third month. End of the third month means here completed first trimester. From here to here first trimester is completed. Once the first trimester is completed, all the major organs are formed. So what about remaining 6 months? remaining 2 trimesters means that 6 months is useful for only the increasing in size and also completion of the organs development. So, at the time of first trimester all major organs are formed. Let us see 5th month. In 5th month we can see movements of the fetus, we can see inside the fetus is going to move limbs and head like that, and another one is hair is also appeared on the head. At the end of 6th month, means the end of 2nd trimester, body is covered with hair. In 5th month ending only on the head we can see the hair, but at the end of 2nd trimester means at the end of 6th month we can see the body which is fully covered with fine hair, and eyelids are separated and eyelashes also we can notice during the end of 2nd trimester. And at the end of 3rd trimester means at the end of 9th month, fully developed fetus with full all characters, we can see it is ready for delivery. So, like that these are the different events. So, like that fully grown fetus is going to get delivered.